normal genes with normal function would be considered the wild type, but changes in the genotype can change the phenotype. Those DNA mutations can be positive, negative, or neutral, which partially depends on the environmental context. Random mutations come about through errors in DNA replication and external factors like mutagens. So when we're talking mutations, again, we're talking about something changing in the DNA. And typically we understand that DNA leads to messenger RNA, which leads to amino acids, which create our proteins. However, we can have point mutations where we have a substitution. One letter of the DNA code is changed, which then has a subsequent change in the protein because the amino acid has been changed from phenylalanine to leucine. You can also have frameshift mutations, which are typically more serious because by inserting a letter, it changes the triplet codon sequence, which doesn't just change one amino acid, but will change every amino acid after the insertion. We also have deletions, which also create frame shifts. So when we think about mutations, a lot of times we think about the DNA changing, but then how does that affect the ultimate protein? So that comes back to this RNA amino acid chart. So let's say, for example, we had a DNA code that created the RNA AUC. That would give us isoleucine. But if a mutation happened in the DNA where instead of getting AUC in the messenger RNA, we got CUC in the messenger RNA, we wouldn't get isoleucine, we'd get leucine. Now, that doesn't seem like such a big deal. And if we imagine another scenario here where instead of AUC we get AUU, we don't even change the amino acid, and it would still be isoleucine even though a mutation had occurred in the DNA. So typically we think of point or substitution mutations as not so serious, but one serious example would create a stop codon. For example, if the DNA sequence was supposed to make tyrosine and it was supposed to be UAC, but instead of UAC we got UAA, that could prematurely stop the creation of that protein. And the average size protein is 400 amino acids. If you're supposed to have a tyrosine 20 amino acids in and it hits a stop codon, instead of being a protein that has 400 amino acids in a very complex three-dimensional shape, you might get a little 20 amino acid protein, which would not be able to have the function of that original protein. So typically when we start thinking about mutations, it's not always environmental mutagens, just errors happen during replication. So during replication, DNA polymerase is adding nucleotides to the original parent strand of DNA. And mistakes happen. But we have other things involved where DNA polymerase will proofread and double check to make sure the letters are correct and can go back and fix them. So typically we think of the error rate in replication as 1 in a billion. So 1 out of every 1 billion nucleotides could be the wrong nucleotide. Now, when we start thinking about this being a numbers game, we think of the number of cells dividing per day. It's about 50 to 70 billion cells you are making new every single day. And in every single one of those cells, there are 3.2 billion base pairs per cell. So you have 160 to 224 quintillion base pairs being made daily. When you take this number divided by this rate, we might expect 160,000 to 224,000 mutations daily just through errors in replication with the creation of new cells. On top of that, environmental factors can increase that rate of mistakes happening. So we call these things mutagens. So chemicals, certain additives in the past, we didn't realize they might have helped, but we didn't realize that they were caused mutations, like benzene. Tobacco smoke has been known for a long time now to be a mutagen, and also various pesticides. We can also have physical things in our environment that can cause mutations, such as X-rays, different types of radiation, and although sunlight and UV radiation is essential for some things like the synthesis of vitamin D, it can also cause DNA damage, which can cause many problems. We also have some biological mutagens, like certain viruses, which can interrupt the checkpoints where the DNA damage is being repaired. There's specific viruses that are known to affect tumor suppressor genes. So oxidative stress is not a mutagen, but it's known and it's linked to many different problems because it can cause DNA damage as well. So through chronic inflammation or simply the incomplete redox reactions involving cellular respiration, we can get these free radicals or reactive oxygen species, which then can cause DNA damage. So when we start thinking about that, we have a natural error rate of 1 in 1 billion. We add mutagens, and now we could increase that rate 10 to 1,000 times. And again, it depends on the exposure and how severe the mutagen is. But now we could take a look at an error rate instead of 1 in 1 billion. That error rate could be 1 in 10 million, or 1 in every 100,000 base pairs could be mutated. Which you might be asking, how come we're not all mutants? And in a way we are. We have changes in our DNA, and we pass some of those changes on. However, most mutations don't change anything about our phenotype. And one way to think about that is, again, this idea that if you only change the last letter of a code, you're still getting the same amino acid. So all of these are isoleucine, all of these are leucine, all of these are proline, all of these are serine, all of these are valine. So we have a lot of redundancy in that last letter. So even though a mutation may occur, it may be silent and not change the structure of the protein. We also have this non-coding DNA. Again, there are like 3.2 billion base pairs per cell, but only 1 to 2% of those are actually codes for proteins. So in order to change the shape of a protein, you would have to mutate only that part of the code that's coding for the proteins. So then what is all that other non-coding stuff? Well, it could be introns, it could be sequences between genes. There's transposons, which are repetitive sequences. But we also consider all of the DNA that codes for the ribosomes, the transfer RNA, the microRNA, and even the small nuclear RNA that involves splicing. All of that would be considered part of this non-coding DNA. The DNA also has promoters and enhancers and other regulatory sequences that are part of non-coding DNA. So when we talk about mutations, again, it's not always so serious because there isn't that much of our DNA that's always coding for proteins. But the fact is that mutations don't only change the structure of the proteins, they can change the expression of genes. So it may change the amount of protein being made and not necessarily the structure of those proteins. So our first example here we're going to look at is this MC1R gene. An MC1R gene makes a protein that's a receptor. And when that receptor is received, it makes melanin. Okay, so in these melanocytes within the skin and hair, if there's a message, the receptor will make black pigments. If there is no activating signal, it might make red pigments or not very much, or just light colored pigments. So the example we're going to look at is first here is pocket mice. So if we have a gene that creates a protein that's not functional because it's been mutated, the gene's been mutated, it's going to make a very light colored mouse. However, if we can also get very dark colored mice by having this protein being turned on a lot. So when we look at this has evolutionary implications because we can have variety in the phenotype based off of what the genotype that's been inherited through mutations. So again, when we start talking about evolution, it comes back to survival of the fittest, which it's not just about surviving, it's about reproducing. So that ties us into the environment. So when we talk about natural selection, it's not a choice, it's the nature that's selecting. So if you put these 4 mice in this environment and these 4 mice in this environment, the environment is basically going to select who survives. Up here, these guys will probably get eaten. Down here, these guys will probably get eaten because they don't camouflage as well. So it's those survivors that will be able to reproduce and pass on their particular genotype because the phenotype was favored by the environment. So again, when we think about this particular gene, we're talking about these pocket mice. However, this same gene exists in all vertebrates. And it's not just every time a light colored mouse shows up, a mutation has occurred. When the initial mutation occurred, could have happened thousands and thousands and millions of years ago. So that mutation ultimately creates variation that can be passed on. So this MC1R gene is almost like an ancient gene because all vertebrates have versions of this gene, and it is responsible for the different color variation in bird plumage. And it creates different variations like in these jaguars. And even whiptail lizards have different color variations based off of this one particular gene. We even see it in humans with hair color. So next example, we're going to talk about cystic fibrosis. And cystic fibrosis is a disease that comes from a chloride ion channel and the gene that makes that channel. So the gene that makes this channel is found on chromosome 7. And if a defective gene, if the gene is defective, it'll make a channel that doesn't allow these chloride ions to pass through. If they get stuck inside here, the respiratory tract and other regions in the body get filled with this thick, sticky mucus. And ultimately, advances in medicine have come a long way recently, but people with cystic fibrosis typically have died at a young age. So, again, if you would be homozygous dominant for the wild type allele, for the protein that works, you would be able to have a normal mucus layer and chloride ions would be able to go through the channels inside and outside. If you have cystic fibrosis, again, the channels don't work and this mucus layer is too sticky. And again, we would think of that as the homozygous recessive, broken, mutated version of that gene. So when we're starting to think about heterozygous, you would still have one functioning chromosome, number 7, that would make healthy, normal channels, the chloride ion channels, but they would only be half as many because that other chromosome isn't making the protein channels correctly. Now, we usually think about this as either you have it or you don't, but again, since we're diploid, half of these are good. And from an evolutionary standpoint, this is actually a benefit because typhoid, cholera, tuberculosis, all the bacteria that create those diseases make toxins that cause these channels to send chloride ions out. So if it's flushing all these extra chloride ions out, that causes a lot of the health impairments that are associated with the consequences of this disease, like diarrhea. So when you only have half as many, you're not losing as many chloride ions, and they're actually more resistant to these diseases. So it's a benefit to be heterozygous. Another example that we can look at, and the last one we'll take a look at here, is the hemoglobin. And hemoglobin is found on the 11th chromosome. And if you have a simple substitution, we could get valine instead of glutamic acid. And that substitution in the protein for hemoglobin creates sickle cell. So, again, coming back to some of these evolutionary implications, if I talk about malaria, you're typically going to think about mosquitoes, but the malaria disease is actually caused by microscopic plasmodiums, and their life cycle includes red blood cells. The mosquito is just the vector that spreads these plasmodiums. So if a mosquito would suck your blood and put these plasmodiums into your body, they would go through one life stage within your liver, and they'd go through their next life stage in your red blood cells. And it's in these red blood cells, when they burst, that causes all of the problems with malaria. And when we think about normal hemoglobin, that would be what we usually think of as dominant. And if you have both broken versions of the hemoglobin protein, you would be said to have sickle cell disease. Now, if you are heterozygous for this, this is actually a major advantage when it comes to malaria. And because this extra life stage that occurs with these little plasmodiums can't occur and be so detrimental if you are heterozygous. You still don't end up with all the clotting and other issues associated with sickle cell disease, but you're a lot less likely to get malaria. And when we start thinking about this again, it's everyone who has sickle cell disease their DNA was not mutated. They inherited that mutation. And the original mutation that began sickle cell, people estimate it's over 7,000 years old. And again, that mutation is going to have evolutionary implications, because the original mutation is actually going to be beneficial, because the people, especially within this region of Africa, would be able to survive malaria and go on to reproduce. As they reproduce, they'd pass on that allele for the mutated version that causes sickle cell. So again, when we're thinking about mutations, they can be positive, negative, or neutral, depending on the effect they have on the protein and the genotype. But also, we have to look at whether they're beneficial or detrimental based on the environment and the ability to survive and reproduce with that mutation. So again, we have normal function, but mutations can be positive, negative, or neutral. And ultimately, those mutations come from either errors in DNA replication or can increase through external factors.